All right. Good morning, everyone. And thank you for coming so early to this amazing session on biotechnology approaches for animal and crop improvement and environmental risk assessment of genetically engineered organisms. My name is Adi Alt-Holland, and I'm a professor and a faculty member at Tufts University in Massachusetts, and I'm honored to co-convene this important and exciting session together with Professor Hong Lu from Clemson University, who is co-convening it with me. This session follows a very insightful and amazing session of panel discussion that we have yesterday morning. It will cover research advances in biotechnology risk assessment, gene containment, and risk mitigation strategy development in both plants and animals. Before we begin and introduce our speakers, I would like to recognize the support from the USDA Biotechnology Risk Assessment Program, or BRAG, that support generation of information that will assist science-based decisions on genetically modified organisms. With that, I would like to introduce our first speaker, Dr. Yiping Chi from University of Maryland, College Park, who will introduce himself and present his talk about investigating off-targeting effects of genome editing technologies in crops. In crops. Thank you very much, and welcome to the podium.
Good morning, everybody. Thank you for coming so early, as Adi said, appreciate that. So today I'm going to give this talk. All right, so today I'm really going to cover off-target analysis of a few major genome editing technology in crops. So as we know, type 2 CRISPR-Cas nuclease and also base editor has been rapidly developed and used in many plant species. For example, here you have Cas9 nuclease, which is RNA-guided DNA nuclease, which can generate DNA double-strand break. And upon DNA double-strand break, it will require DNA repair pathway like NHEJ to make, sticking DNA back again that may be error-prone, which may introduce mutation. That is, many of us are using this feature to knock out genes. If you are to provide a donor which can allow HDR, homology direct repair to happen, then you can store very precise genome editing outcomes. So of course, after CRISPR-Cas9, there's more CRISPR-Cas system being developed, such as Cas12a and also Cas12b. But nevertheless, all these CRISPR-Cas tools, they are all type 2 CRISPR-Cas nucleus, which meaning they are really requiring only a single Cas nucleus to do the gene editing, which is very convenient. That's why many of us are using this kind of technology. Let's go back to CRISPR-Cas9. So what's really interesting about CRISPR-Cas9 is it is actually a very versatile nucleus where we can mutate one of the nucleus domain so that it still can function as a nucleus but can only cut one strand of DNA. This is called a Cas9 nickase. So when you have this Cas9 nickase Cas9, you can actually convert to or develop more tools such as base editor. So in this case, I show you in the screen is where we can fuse a deaminase, specifically cytidine deaminase, where it can convert a C2U mutation, DNA repair pathway, or replication will fix that U to T so that you can actually achieve C2T base editing. So if you are to replace this cytidine deaminase with a adenine deaminase, you're going to achieve A to G base editing. So this is really the key core toolbox in the base editing toolbox. So today I'm really going to cover the off-targeting analysis in a genome-wide scale in crop plants. So, but I just want to remind you what really I'm going to focus on today. So I'm going to focus on guide RNA independent of target effects of Cas nuclease and base editors. Because when we talk about off-targeting, it can cover many things. Just like you design PCR primer, you made, if you don't pay attention, not using better software, you may end up picking an oligo or a primer which are not very specific, right? It can adhere to many loci in the genome, and you complain, oh, my PCR product, I have so many non-specific amplification. We define those are user sort of related or guide RNA dependent sort of things. So I'm not going to cover all those kind of off-targeting because those are really user dependent and can be improved, right? So I'm talking about really is kind of inherent off-targeting, potential off-targeting feature of these nucleases, which are really not dependent on guide RNA that you design, all right? So I'm going to cover mostly our research in the model crop plant, which is in rice. I have some other data showing experiment also in tomato and also in maize. All these are really major crops. Okay. So let's start the first batch of paper we published really quite a few years ago in 2018, sort of back to different journals reporting these genome scale of targeting analysis. So first one I want to bring up is a publication in PBJ, which is really led by Dr. Ken Wang's group with collaboration with my lab, also Keith Jones' lab in Harvard University. So in this case, this study is really being led by my good friend Keng Song Lee. So where we actually using a very different approach to assess sort of genome and off-targeting effect. So in this case, we are really using making a CRISPR-Cas RNP in vitro, and then we extract DNA from maize, and we digest the maize genomic DNA and in vitro so that we can sort of probe where the DNA break has been induced in genomic DNA in the maize, right? So this methodology is going to help us to nominate potential off-targeting site. And then we go after these off-targeting site by really using a genome-edited maize plant resulting from same reagent using stable transformation. And then where we did it is basically doing amplicon sequencing to really go after all these top potential off-targeting sites to see where's really in those edited plants, can we find any mutation on those off-target sites? And the answer is no. Okay, this is as much as we can do at the time with our budget, because as you know, maize genome is very complex and big. If you want to do whole genome sequencing, it's very costly, not to mention that a lot of reported sequence transposing in the genome may actually prevent you doing a very thorough genomic analysis anyway. However, in second example I show you here, below in the slide, is actually in rice. It has rather a small genome, only a short of actually 500 million base pair. That means we can actually use whole genome sequencing to really evaluate off-targeting effect very comprehensively. So in this study is really collaboration with two scientists in China, in the Yong Zhang and also Tao Zhang lab. So where we did is we really are generating genome-edited plants by both Cas9 and Cas12a, and then we by different guide RNA, we also generate plants without any guide, just Cas protein expressing that. Then we look at whether we can find any off-targeting effect. And this thorough analysis also reveals that there's barely any evidence on guide RNA independent of targeting mutations. So which are really, really encouraging. So then we further move on to test this in different Cas nucleases, in this case Cas12b, where we also published it a few years ago as a very useful tool for genome editing. So and relatively recently, a few years back, we also applied genome sort of wide whole genome sequencing approach to look at off-targeting effect on Cas12b. So on the left panel A, as you can see, we look at how many single nuclear variation SNV discovered in the wild type, which is just really wild type plants we use, the material we use for genome editing. That number of mutation recovered from wild type just really means the different mutation, the difference between our crop versus the reference genome. And then you also have GFP as a transformation control. So the mutation we recover from GFP tell us the level of somoclonal variations, right? So, and then you have more, we also have a Cas12b backbone, meaning we only have Cas12b expressed without using any guide RNA in the construct. So if there's any guide RNA independent of target mutation, we are expect to see that also we can find those mutations in this only Cas12b construct. Of course, we also have Cas12b construct with guide RNA, which are typical usage. So anyway, on the A panel shows you how many SNV discovered, the B panel shows how many India discovered. So as a quick glance, you can see we didn't really find any statistical significance among all the mutations discovered. As a good indication, there's really no genome-wide off-target mutation discovered for Cas12b. And just a few months ago, so our patent application for using Cas12b in plant has been approved. If you're interested in using this technology, you can come talk to me. All right, so but you may wonder, how can we really do sort of targeting analysis? I just want to show you this pipeline first before we dive into more data. So because this is a general pipeline we've been using, I'm going to talk about over and over again today in this talk. So in panel A here, it just shows you we will be generating stable transgenic plants which are genome edited, T0 generation, let's say. And then we will group them in two classes. One class is really genome edited plants. Many of us are really interested in pursue those, editing outcome. But because this is really for analysis of targeting effect, we try to include many controls, right? So one control could be just wild-type plants. We sequence that to review any background mutations. And also, we can also have a Cas-only control without any guide. And then we also have Cas plus guide RNA, right? So if we expect any of mutation will result from off-targeting effect, what that is guide RNA independent, then we will expect that both Cas protein only and also Cas plus guide RNA will result those kind of off-target mutations. So then B panel shows you the pipeline, a computational pipeline, where this is still we are using Illumina sort of short read sequencing, but you can imagine similar pipeline can be established for long read sequencing. So basically for both SNV discovery and also in their discovery, we try to use three software to do the analysis and then look at shared outcome and go forward. So which give us more robust sort of analysis. So so if you have more questions, something you can come talk to me after this. I'm not going to delve too much into detail about the pipeline, but I just want to show you here. All right, so I just want to quickly summarize what I just showed you, our conclusion here. So basically, SP Cas9, ALB Cas12a, as well as AA Cas12b that we evaluated, we didn't find any off-targeting mutation. This is very simple, so question. But these kind of results are already very encouraging, right? So I remember, so I was pretty excited realizing the Secure Lua released in 2018 that actually two of the major papers I just showed you being cited, as a scientific evidence for off-target analysis sort of outcome by CRISPR-Cas system. Because before our work, there's really no genome-wide analysis available in a peer-reviewed literature. So I think it's very important as an independent researcher we provide us scientific evidence for that. So even though we know we are rolling back to the Lua before Secure Lua, but overall this kind of scientific evidence never gonna go away, right? So this is taken from a review paper that we wrote 2 years ago, and which really shows you the global map of regulation. So it's very colorful, right? But in this case, colorful is really, I don't know if it's a good thing or not, but it basically just tells you a very complex a situation in different countries in terms of regulation. So I don't even want to get into detail about outcome of genome editing, whether SDN 1, 2, 3 is getting even more complicated. So here the regulation are really concerning about making small insertion deletion without bringing any sort of DNA sequence from a foreign genome, right? So just very simple genome editing outcome. This is regulation map right now. So US and South America, many countries, as you can see, still relatively very friendly, also Canada. But if you go to Europe, it's rather, it's complex. So things are moving along, but very slowly. We still don't know, even there's a rule, being, pushed forward by the EU, but we know independent member country, how they enforce that is going to be a question. So that's going to be much, much complicated situation. So nevertheless, many countries try to catch up with all the technology being developed rapidly in the field of genome editing, and we'll see, how things are going to pan out in the future. So this is a pretty only slide I'll show you about regulation because I'm not that interested in that. So anyway, we already covered it yesterday, so I'm going to really show you more evidence about, science. For us, I think what I can do is really to provide scientific evidence, hopefully, to really guide or provide, many regulatory entity bodies to use those information and for any of regulatory direction. So right, so I'm just kind of joked, it's important data, right? So I showed you with single guide RNA we didn't find any of mutation, but the question quickly comes. Same thing for regulation. People already asked in yesterday's panel discussion, what about I'm introducing many or multiple mutations in the genome? What happened, right? So because genome editing tool has been touted as a very useful tool for accelerated breeding, meaning if we know the traits linked to a mutation, we can simultaneously introduce those traits together to really speed up the breeding process, it is really great promise for genome editing technology. So the idea I show you, or the idea I show you here is really de novo domestication, right? So you can imagine that 3 mutations, you can introduce in different chromosomes, in this case in tomato, let's say, or cherry. So if you bring them together, right, so you have all the traits within 1 generation, ideally. But however, in this case, you are generating multiple DNA breaks in the genome. Is the DNA repair outcome going to be the same? Are you going to see more mutations? You can have your hypothesis. So this is really a question we want to kind of try to answer. So in this case, we try to focus on CRISPR-Cas12a nuclease system. So a few years back, in 2021, we published this paper in Nature Communication showing a very, very robust multiplex genome editing system based on Cas12a. And we, on the very left, which I highlighted, which is a system which are basically using a dual POR2 promoter, driving both Cas12a and also guide RNA, multiple guide RNA. And we are also using the HH and HDV ribozyme system for precise processing of the guides. So this really results very efficient editing in crops. So then we use this system to test if we are to introduce many mutations, many double-strand break in a genome of rice in this case, what's going to be the outcome? So we have done 2 scenarios, 2 cases here. In the case 1, we really induce limited double-strand break using 4 guide RNA. We actually try 2 different Cas12a nucleases. One is ALB, another one is MB2 Cas12a. So in second case, what we did, we tried to induce many DNA double-strand as we can with our construct, right? So in that case, we are using 16 different guides to target 27 genomic target sites, which I kind of illustrated in the chromosome map top in the slide. So where you can see almost every chromosome is being targeted, all out of 12 chromosomes in rice being targeted by this single construct. So the question is very interesting, right? Can we find more mutations? If so, is that an indication of targeting effects? So anyway, so on the lower right corner, I show you the data of, again, revealed by whole genome sequencing of these rice plant about SNV number and also indel number. So as a quick glance, you may see that on the left, the SNV, well, compared to the tissue culture only control, that's really not many more mutation or SNV being produced, right? So of course, we can also go after doing a more detailed, thorough analysis of those mutations just to see whether there's any indication which is guide RNA dependent. In some cases, yes, there are some mutations indeed seem to be guide RNA dependent, which meaning those mutations can be predicted by like Cas9 Finder, those software. On the right, you can find the indel mutation. So you can find maybe one a group of construct kind of have more mutation, but that's really even though it's maybe that's significant, but that's really, we review that, investigate that really appear to be still a tissue culture result, somoclonal variation. So what's really interesting is when we look at the construct, even with 16 guide RNA, we didn't even reveal many more mutation compared to lower multiplexed construct or even the control, which is again a good sign. Maybe there's not many off-targeting mutation induced even with so many DNA breaks in the genome. Well, let's look at on-target mutation. So this is a view of all the 27 mutation or all the target sites, right? So you can see in some plants, we really can get edits like for 23, 20 edits at these sites. It's really multiplex editing has been revealed on these on-target sites. So these are the plants we try to really investigate. Is there any interesting, weird, chromosome arrangement due to these, double-strand break happening in the genome? And the answer is actually yes. So you have to invest very carefully. I just want to give you 2 examples. So in one plant here, where we find is what's really interesting is there's actually, this is in rice. It has 2 copies of chromosome genome, and in one case, one allele we have found is a very small insertion in the 9 base pair deletion, but another allele we found is actually more than 1.5 kb, large deletion. So I say it's kind of interesting because typically we don't really discover this big deletion with just a few guide RNA, but in this case it happened. One hypothesis is because you are having so many nucleus targeting DNA in the genome, right? So you can imagine the DNA repair pathway is being saturated or tired, try to rush to repair all the double-strand break. So there may be some delay, in the repair process, which may result in more extensive excision of DNA at certain target site. This is one example here, right? So In another example I can show you is even more interesting, where we found actually one just DNA region close to a target site T12, we found about 1.3 kb DNA sequence has been duplicated, which means being actually inserted into chromosome 3 at another target site. Which again give you some scenario you can imagine when you have so many DNA breaks and then try to repair. They try to sometimes use template-based repair. So then they somehow capture some other DNA sequence, try to insert and fill in the gap, the break site. This is really one case, very interesting, so we discovered. So we didn't discover any of these kind of chromosome rearrangement in the plants result from multiplex editing of only 4 guide RNA. We didn't find all these events actually are discovered in those plants during many DNA breaks. All right, so I just want to give you a summary. So you can follow me. So this is very simple for this first part. Our second part, actually, so on multiplex editing. So our conclusion is simultaneous introduction of fewer, let's say about less than 10 DNA breaks per cell of DNA breaks by Cas12a are unlikely to cause chromosome rearrangement. So I try to be very careful, right? Because we only evaluated in rice, we only evaluated in limited examples here. So however, simultaneous introduction of many, let's say even more than 50 double-strand break per cell, can lead to unpredictable chromosome rearrangement. When I say unpredictable, because those things I can never predict how large the deletion will be, where the DNA will be replicated, inserting to another location, rather hard to predict. So so that's really defined outcome if you are thinking about the level of multiplex editing. So this is really one take-home message you have. Think about when you're designing your experiment, conducting multiplex editing. So the more targets that you have in the genome, the better chance you actually will have, the higher chance you'll have these kind of chromosome rearrangement as a interesting off-target outcome. Okay, so now I'm going to move into discuss off-target effects by a base editor. Here, specifically cytosine base editor CBE and also adenine base editor ABE. So some of you may not really follow closely with the deaminase mechanism. I just quickly introduce that, walk through with you. So as I mentioned, Cas9 can be converted to a nucleus, sort of a nicking nucleus with one mutation. So in this case, we can mutate Cas9, make a Cas9 D10A mutation with just one mutation, where it will only basically cut its targeting strand. So the non-targeting strand, it won't be cut at all. So then we can fuse a cytidine deaminase. In this case, I show you from, let's say, a mouse upper back protein fused to the N-terminal, in this case of Cas protein. And then we also fuse another enzyme called UGI, which is uracil glycosylation inhibitor. So this is actually very critical. If we fuse the UGI to this system, we can actually achieve very pure C2T base editing. So on the right, it's really illustrated the mechanism where it happened. So where you have C converted to U by the deaminase, and then the U is being not removed because we have a UGI, uracil glycosylation inhibitor there, right? So you try to keep the U there as long as possible, so giving time that DNA repair or DNA replication will happen to capture that information and recognize U as a T. So that's why in the end, you're going to be fixing from C to U a mutation. So this is how C2T base editor works. Of course, there are so many cytidine deaminases to be explored for C2T based editing. So we have done, in this paper, we have done quite many of them. I just want to show you 4 of that, which is HAID and also A3A, Y30F. So both of them are from human. And also then rat upper back 1, and also then PMCDA1, which is from fish. So all 3, as you can see, we test only 4 of them. We use these CB, let's say, let me back off here. So in this case, the CB we are using is specifically configured as a BE3 sort of configuration, where you will have a deaminase and UGI fused to the Cas protein. This we call that BE3. So if I say BE3, you kind of have a good idea what that means. All right, so all these deaminases were configured into a B3 configuration, and then we generated transgenic rice plants, and also with sort of guide RNA only, deaminase only, and also deaminase with guide RNA. Those all kind of control, as I showed you earlier. So then we look at how many SNV we discovered in individual plants and how many indels we discovered, right? So I didn't show you the data. Basically, we didn't find significantly more, SNV or indels in our edited plants. However, in the plant result from HAID deaminase, we indeed find a significant number of more mutations. And we actually found many of the mutations indeed are C2T mutation. So I just only show you this slide as a further analysis here. So you can see the highlighted two windows there, which are really summarizing the C2T mutation by 4 deaminase. So as you can see, some of them really give you many more proportion of C2T mutation, suggesting there's really off-target effects. Okay, so some of them are, bar is very comparable to the control, meaning there's really barely any C2T mutation discovered in those genome edited plants. So which is actually happened to the PMCDA1. We cannot detect any this kind of mutation for the plants result from B3 based on PMCDA1. Okay, so this is basically based on these mutation, we ranked, which one has more off-targeting effect. This basically HAID has more than A3AY30F, and that is more than rat alpha-1, and then more than the PMCDA1. So the PMCDA1 is really the most precise base editor if you want to use. However, because we didn't really discover so many C2T mutation for the plants resulting from A3AY30F deaminase, we actually found that this deaminase is very useful because it has very sweet editing window. So we then move ahead to use this deaminase to do a few examples of genome editing, because some of you may wonder why this is so important, right? So because editing deaminase, it can make C2T mutation, meaning it can introduce stop codons. We actually can, instead of using Cas9, Cas12a, this kind of nucleus to knock out gene, we actually can use deaminase to more precisely introduce stop codons to knock out gene. So one example I show you here is where we did a multiplex editing of 3 target sites in the rice genome. And in fact, within 1 generation, it's so efficient, in the T0 generation, we can generate homozygous plants at all 3 sites. Can you imagine that will happen for Cas9 or Cas12a nucleus? It barely can happen, right? Because all the repair, NHD outcome is rather hard to predict. So that's why this is useful, because you can really control the editing outcome in a very precise fashion. Because it is so useful, we actually developed a tool called CRISPR-Bets to guide a user to design guide RNA to precisely introduce stop codon to knock out your gene of interest very efficiently. And the name is called CRISPR-Bets. So that's 2 meanings for bets. Bets means base editing to stop. But it also means it will give user best bets to find the best guide RNA to knock out your gene, all right? Okay, so second example I want to show you is really another powerful application of this kind of base editor, where you can really design a family, a group of guide RNA to target a region of a gene, which, to introduce sort of relative random mutations. So then you can select the outcome, looking for any of the outcome of altered protein structure, etc., which result in gain of function. In this case, is herbicide resistance, right? So in this case, we actually targeted the AS1 gene, and then grow the doing tissue culture using selection, and then looking at resistant plant. This allows us to find the novel mutation which actually confer herbicide resistance in this gene. Okay, so move on to show you more data on off-targeting analysis. I'm going to move away from rice now because we want to target another species to get some general ideas. In this case, we collaborated with Joyce Van Eck group at the BTI. So in tomato, we also use this very efficient A3Y30F-based B3 to edit tomato gene, and indeed we find the efficiency is pretty high. And then we did a whole genome sequencing of these plants. So as you can see on the left, there's SNV. There's some level of more mutation, although it's not statistically significant. But we do, there's higher level of the mutation. Some of them are C2T. May indicate that slight some off-targeting, but it's again not statistically significant. We cannot conclude that's really off-targeting effect being detected in this case. But for the indel, that's actually not much at all. It's all comparable. So we also look at RNA level, because all these deaminases, how they function is they actually act upon single-strand DNA. So that's a good chance they can also act upon single-strand RNA, right? RNA is single-strand anyway. So that's a chance they can edit RNA. So that's why we did whole genome sort of RNA-seq to look at in any mutation at RNA level. The short answer is no. We did all kind of analysis I showed you. I don't want to further dive into that, but you have trust me, the paper published. So it's actually we didn't find any evidence at RNA level mutation. So we still continue, my lab still continue to do a lot of sort of discovery on the novel deaminase, hopefully with a better feature for base editing, in this case C2D base editing. So these three students work together, actually 2 graduate students, 1 undergrad student, did a phenomenal job over the years, and they have actually screened 66 novel deaminase, mostly sourced from the animal kingdom, and then really rapidly assess the activity, and then use stable transformation rice to further probe that features for introducing C2T-based editing. So we really interesting thing, we find quite a few of them are really, really efficient base editing tool. So a few of the deaminase discovered is very interesting. They all come from ocean mammals. The really outperforming one is actually from killer whale. So, and the deaminase name is here is 0084GX2. So I just show you one slide here. We just published recently in this paper. So we found this deaminase-based CB really can confer very robust multiplex editing in rice. The reason being this deaminase is relatively less context dependent. If you look at the first figure panel, I kind of highlighted with 2 windows. On the right one is the one A3A-Y30F. I just showed you a bunch of data already. So it actually very much prefer a TA context, meaning if that C is preceded by other nucleotide, the chance of editing is kind of relatively low. So that kind of prevent it to achieve very universal high editing efficiency. However, the one I highlighted on the left is have reduced TC context. Just because this feature, it actually enables more robust editing across many target sets we design. For instance, again, I highlighted in the second panel on the right top. So where we find this deaminase from killer whale enabled highly efficient at this target set where otherwise the A3A-Y30F barrier can edit. Because that reason, we can achieve very efficient editing with this new deaminase. And we also did a whole genome sequencing, didn't find a very alarming, off-targeting effect at all. Just the off-targeting effect is very much similar to the A3A-Y30F. Only a few C2T mutations appear to be off-targeting, but it's not really high. So okay, so let me conclude this part. So I showed you PMCDA1 based CB is actually a very efficient and also a deaminase with no off-target effect detected in rice at all. Okay, so another deaminase A3A-Y30F based CB is very efficient with some off-targeting effects detecting rice, but again, not in tomato in both genome and also at the transcriptome scales. So also AAA3GX2 from killer whale is a very robust deaminase suitable for multiplex genome editing. So finally, I would say off-target effects of CB are dependent of plant species and also maybe transgene expression level. So I'm now going to be diving to the last example quickly. So I already showed you how CB works. I don't want to explain too much. In this case, this AB works is you basically fuse the adenine deaminase to a Cas nuclease, then which will induce A2G base editing. And this is first version has been demonstrated in humans again from David Liu's lab in Harvard. So it's very, very powerful, but later on they actually even make better. So you use evolution to identify a very efficient sort of AB8E deaminase, highly efficient for base editing, introduce A2G mutations. And because it's highly efficient, we actually fuse this deaminase to a Cas9 variant called SPRY. So this SPRY is nearly PAMless, meaning it can almost target anywhere in the genome without dependent on the NGG PAM typically you would have for SP-Cas9. However, the trade-off is that this nuclease typically is of low efficiency. But because this AB is so efficient, we decided to fuse to this SPRY to achieve PAMless base editing. And we were able to show, in this case, in plot find activities. And in stable plants, we can recover efficiently these A2G outcomes. Okay, so then we do whole genome sequencing analysis, and find it's very interesting. So meaning the plants, as long as they express this AB80, it doesn't really matter with guide RNA or without guide RNA, we discover many sort of actually a mutation, which is A2G mutation. And then we look at the mutation, and then there's actually a TA motif, meaning if that A is preceded by a T, that A is highly likely to be edited in the genome. So when I say this genome off-targeting effect, it can go to almost nearly like from 500, even up to 1,000 mutation, depending on how high expression level of the deaminase is. If you have low expression deaminase, the mutation is much lower. So again, it's not that alarming, but you have to be careful. This deaminase indeed can introduce mutations, and this is very much collaborated with almost back-to-back report from a Chinese group in China, from Huanping Zhou's lab in China, where they actually did RNA sequencing from rice plant result from ABAT. They also discover at RNA level there's also sort of TA motif being preferred. So again, collaborating with our close collaborator Joyce Vanek, and we also applied ABAT to do base editing. In tomato, we showed high efficiency base editing, and then we also used the resulting plants and did a whole genome sequencing. And in this case, interestingly, we actually didn't find our base edited plants compared to the tissue culture only control plants. That's really not statistically significant more mutations, which is really a contrast with the rice data, suggesting we didn't really find a good evidence of off-targeting mutation at DNA level. But then we want to make sure, what about RNA? So we also did RNA-seq, and we for all the mutation we recorded from at RNA level, we tried to look at is there any motif signature like TA motif at all? The answer is no. As you can see, there's really no TA motif stand out for any of the mutation we discovered, suggesting indeed this deaminase somehow is very precise introduced mutation in tomato. Just to summarize this part, so ABAT is highly efficient in rice and tomato, and ABAT has detectable off-targeting mutation at TA motifs in rice, but there's no detectable off-targeting mutation by ABAT in tomato in both genome and transcriptome level. So finally, I just want to reiterate this key sort of conclusion. So off-targeting effects of base editor are dependent on plant species and also, again, transgene expression level. So my lab will continue doing this kind of research to provide a lot of peer-reviewed paper so that regulatory agency can look at the data to make their more rational decision on regulation. So in this conference, excitingly, two of my graduate students were actually presenting two very new, exciting CRISPR system. Josh going to talk on Monday, going to talk about a new type 1F CRISPR system for generating larger deletions. And it's really new. Nobody even published this kind of work in human. This is really de novo discovered new CRISPR system. So, and Innocent is going to talk about a CRISPR combo system for simultaneous genome editing and gene activation. So we are also interested in doing whole genome sequencing to look at genome-wide off-targeting mutation with these new systems. So with that, I just really want to acknowledge those people in my lab doing hard work. I'm really glad, fortunate to have these very young, energetic, exciting scientists working in the lab. And then I also want to acknowledge collaborations from these people I already mentioned. So finally, importantly, I really acknowledge the support from over the years from NSF and also USDA, and specifically 3 USDA BRAG grants supporting us to do whole genome-wide sort of analysis on off-targeting mutations. So with that, I'm going to conclude here to answer any questions. Thank you very much. I'm not sure I have time for questions.
Hey, Evening, nice talk. The question is that PMCD-A1, you said no off-target mutations. Can you explain again why that's so specific? And also, does that affect the efficacy?
Yeah, so this is very interesting. Deaminase, that's because of that PMCD-A1, as I said, it's sourced from a fish, and efficiency is quite comparable to the A3A Y30F we are using. However, it can only edit the 5' end of the guide RNA you design. The window is like from 1 to 5 nucleotide. The A3A Y30F has a window of 4 to 10, 12. So it's shifting window. In many cases, we would have a window in the middle of the guide RNA. But if your target happen to be in the 5' end of the C mutation, you want to make it to T is on the 5' end of the guide RNA, you actually can use PMCD-A1. So we didn't really use that very, very often, just just some kind of routine practice because we want to focus on one deaminase. But PMCD1 is very good. As I showed you, we couldn't find any mutation using whole genome sequencing, while other deaminases, more or less, we can find some evidence of C2T mutation, which is not guide RNA dependent. So because of that, that encourages us to search for more deaminases in fish. That's why we go to oceans, things like that. So yeah. Okay, one more question.
Okay, thank you. So I have two related questions. I hope they were quick. One is, do you see, like compared to the tissue culture control, I assume that the mutations there are random, whereas in your off-targets, do you see the same mutations occurring in multiple independent transgenic lines? And then the second related question is, if you are working with a different plant species, like let's say it has a large genome, how concerned do you have to be about the off-targets, or would it be sufficient just to use multiple independent transgenics and assuming that the background mutations would be different in those to prove that your directed mutation is actually causal for the phenotype you see? Okay, thank you.
Okay, try to be quick. Okay, first, for your first question, yeah, that's a good one. So, generally speaking, we didn't find any of the those somoclonal variation always vary among individual plants. There's really no feature; it's just random during tissue culture. So, and similarly, that's we consider somoclonal variation. But for the mutation that occur again and again in our genome editing sample are really good indication. Those are guide RNA dependent. If you look at those mutations, indeed we can find a feature. The target site in fact share some homology with our guide RNA. So most likely that's a result from guide RNA dependent off-targeting effect for those mutations, okay? So for your second question, it's also very good about what if we target like polyploids or more complex genome, what's gonna be different from our example here in rice and tomato, which is relatively small, simple genome. So I really didn't do any experiment, I can only guess here. So I couldn't imagine fundamentally there would be a different DNA repair pathway to function in those species. So overall, I would say it's less worrisome in general with all the tools I just described, unless you're really doing many multiplex editing with so many DNA breaks. Of course, then even you introduce one break sometimes it's in the hexaploided wheat, and then there's 3 ABD genome. Even you're targeting one homolog, you're going to be maybe likely to edit other homologs. That way, maybe some chromosome rearrangement may happen, right? So I'm not even talking about expanding to many independent target sites. So this is a legitimate concern if you are doing multiplex editing. So if the sequencing become cheaper and cheaper, I would encourage if you really develop a product. So using whole genome-wide analysis would be necessary. So, all right, thank you. We can talk later. Yeah.
Thank you, Dr. Chi, for an amazing lecture, and we'll give some time at the end for more questions to come after the 3 speakers will complete their lectures. I'm pleased to introduce Dr. Hong Lu to present his lecture on molecular strategies for transgene containment and removal in plants. Welcome.
Sure, thank you. So, thanks for the opportunity to discuss this important topic. So, today I'm going to just talk about this molecular strategy we have been trying to develop in transgene containment, especially in perennial grass species. Okay. So as we all are aware of the current situation, GM adoptions, not only in the US, but also globally, right? This is not only reflected by the number of crops that has been adopted in modern agriculture, but also by the countries that has been adopting so-called GM products developed by an agricultural biotechnology approach, right? With the development of so-called GMO, if you want, genetically modified organisms, so especially with Yiping just presented excellent progress in genome editing area. So genome editing provides an excellent toolbox for new technologies that will develop the transgenic products not necessarily involved in the transgene integration into the host genome, but in a lot of other cases, transgene integration into the target species is still an effective approach for trait modifications. And that's the case for a lot of crop species. But with that, it raises indeed the question about so-called biotech risk, right? One of the important questions is so-called, what about the transgene escaping into the environment or untargeted species? And this is especially true in perennial species, especially grass species. For example, a lot of forage species and turf grass as a big industry, as well as the perennial grasses that for bioenergy purpose, right? So a lot of them are just weeds. So what if the intercrossing occurs very frequently? So what if a herbicide-resistant genes escaping into the weeds or other untargeted species becoming super weeds, out of control? So all those are realistic concern, about the environmental risk and also the possible for the risk for human health, right? Another big question is like, okay, not only the gene escaping into the untargeted species or into the environment, but also in the final product, a lot of time transgenic technology not only introduce the gene of interest, but also unnecessary so-called DNA fragments, for example, cell marker gene for plant transformation, as well as plasmid backbone and all those. So ideally, so for a so-called ideal transgenic products, you don't want to really have those unnecessary fragments. You would have some strategies in place, not only the targeting, but also the gene containment strategy, so that will prevent transgene escaping into the environment or on targeted species. At the same time, you would like to develop so-called ideal GM products with only the target gene integrated without any unnecessary components. Okay. So, what strategies that we can use for transgene containment? Naturally, so we'll think about in plants, right? Either male sterility or total sterility will be an effective way for, at least in the male sterility case, we can prevent transgene escaping into the wild by pollen dispersion, right? To do that, actually, and there are different strategies, of course, we can seek for to develop male sterilities. So in perennial grass species, we have trying to clone the genes that are involved in anther or pollen development. So this way, by manipulating the genes that are involved in the pollen or anther development, we could achieve probably male sterility and achieving gene containment purpose. So for that, one of the examples we have developed, for example, we seek for the genes in rice, which is specifically expressed in anther or in the gamete, male gametes. So in this particular case, the gene RTS from rice is specifically expressed in the panicle. In situ hybridization indicate that it's only expressed in the tabatum cells, right? And then to test its involvement in, let's see, okay. In controlling plant or anther development, pollen development, we made a construct at the early stage, right? It's the antisense and using its own promoter, which is driving a tissue-specific expression of this antisense RTS gene, as well as a ribonuclease and barnase genes, with the bar as a selective marker gene. So here's shown the example of the transgenic plants in which the RTS gene has been suppressed, and then we can see the pollen anther development is damaged and also resulting in infertile sterile pollens. Okay, so this is one of the strategies that we have used to identify the genes that are involved in the pollen development to achieve male sterility. And the same strategy we use in representative perennial grass species, an important cool season turf grass, creeping bend grass, in which we have been introducing the similar constructs in the transgenic plants, achieving the male sterility. So if you compare the wild type and the male sterile lines, in which the anther development is destroyed. Okay, so so those transgenic creeping grass lines, which are male sterile, has been also tested in pollen cage study in greenhouse, as well as in the field trial under the USDA APHIS permit. Okay, so this table kind of looks kind of busy, but I would like just direct you to focus those highlighted parts. So creeping bend grass is a cool season grass which needs vernalization to be flowering, and also it's a cross-pollinated plants. So when you self those plants, like in wild type control, there is no seeds produced. But if you cross different individuals, and you have the seeds produced. And for all those transgenic lines which are male sterile, when they are selfing, there is no pollen or no seeds produced. Well, even the intercross between the male sterile lines, there is no seed produced. But if you cross with the wild type plants, then you do have the seed production. So this pollen cage experiment indicates the effectiveness of these technologies to induce male sterility, achieving to preventing transgenic escaping by pollen dispersion. And the same approach that we mentioned, that the field trial studies confirm these results. Okay, so this is one of the strategies for achieving the preventing the transgenic escaping by pollen dispersion. Then you may want, of course, in the perennial grass, in the pollen dispersion is one aspect, but what about seed dispersion? Because they could still be pollinated by wild type pollen, and the transgene could still be dispersed by seed, and all those. So now total sterility is another strategy which is actually feasible in vegetatively propagated perennial species, especially in turf species. Okay, so to achieve total sterility, we actually work on a specific gene, the florleafy, which is a homeotic genes, spatial transcription factor that control the transition from vegetative growth to reproductive growth. By using the RNA interference technologies, we actually suppress or destroy the gene expression of the florleafy homologue in creeping bend grass. After vernalization, you can see the control wild type plants flowering normally, while the transgenic plants in which the florleafy gene has been suppressed do not flower. So this again achieved the total sterility. So from those examples that show using molecular strategies is possible to achieve either male sterility or total sterility as a strategy for preventing transgene escape. Now, another question is, so we have the strategies in place that could be used for preventing transgene escape, but how can we implement this into the practical situation and into the final product, the GMO product for commercialization? Okay, so another question associated with this is that, okay, in the final product, how can we just keep the gene of interest without any unnecessary components, like cell marker genes and plasmid backbone, etc. Okay, so for that, you see, so we are using develop a so-called dual site-specific recombination system for control of transgene containment and also the removal of unnecessary genes. Before I get into the specific system itself, I would like to just briefly go over the site-specific recombination system. So site-specific recombination system involves recombinases that are recognize specific target sites, okay? So which do not, are not found in the plants, but are existing in either yeast or different bacteria. For example, flip FRT system in yeast, lock system in E. coli, PHP1, okay? And other systems as well. Okay. Taking one example, like flip FRT from yeast, the recombinase actually recognize a specific target site, which is only like 34 to 48 base pairs, which is specific structure of target sites surrounding a space region, which will determine the direction of the target sites. So in the presence of those two sites, okay, the flip recombinase will catalyze the existing recombination to remove the fragment in between of those two target sites. When those two target sites are in reverse orientation, then the flip recombinase will catalyze reversional recombination to flip over the fragment in between. So using these characteristics, we have developed a system so-called dual site-specific recombination system using two different types of site-specific recombination systems. Specifically, here is flip FRT from yeast and Krilox from E. coli. So we made two different constructs. The first one in which we have a constitutive promoter driving a gene, the male sterility gene or total sterility gene, that will induce male sterility. But they are separated by a stopper fragment in which we have a we had, okay, I can see here. So we have a recombinase for the Krilox system Kri, which is adjacent to the UBI promoter, which would be expressed, and followed by a 35S driving bar genes, which can be used for cerebral marker as a cerebral marker gene for plant transformation. And then we also added an inducible promoter after that, which would drive actually theoretically downstream male sterility or total sterility genes. And this whole fragment, stopper fragment, is flanked by the target sites of another recombinase flip. Okay, so the first construct. The second construct we have, again, a constitutive promoter driving a gene of interest, but separated by a stopper fragment that is composed of the bar gene, or the cerebral marker gene, adjacent to UBI, and also by the second recombinase flip. And the bar gene is flanked by the Kri-target site locks, and FLP gene is flanked by the flip recombinase target site FRT itself. Okay, so those two plasmids could be introduced into plant to generate two parental lines, and they can be propagated by seed production in contained conditions. And that when we cross those two lines in the fourth generation, not brought two constructs together, then the function of the Kri, okay, Kri is driving by the UBI promoter in the first construct, which will then recognize the target site blocks P, not flanking the bar gene, which will remove the bar gene. Okay. And because of the removed bar gene, they'll bring together the upstream ubiquitin promoter to the downstream recombinase flip. So flip will be expressed that not only remove its own coding sequence, because its own coding sequence is flanked by its target site, FRT, but also the stopper fragment in the first construct, the whole thing, not bring together the upstream promoter to the downstream male sterility, total sterility genes. So in the end, you will have the gene that will induce the male sterility or total sterility, and also you have your gene of interest expressed. All the unnecessary DNA fragment, including the recombinants themselves, will be removed. So with that, actually, we have first tested this strategy by using the reporter gene, GAS, to replace the male sterility or the gene of interest. And this system were tested in Arabidopsis and creeping bentgrass as well. As you can see here, the inducible promoter there actually serve as a backup plan. In case the recombination system does not work, then it will drive the expression of male-sterility total sterility gene expression. So indeed, we use a heat shock promoter to drive a gas gene, and that is when this the gas can be induced to be expressed. And then we made 2 parental lines with those 2 constructs. And then those two lines were crossed to produce a F1 generation. And so with the expected outcome, by molecular analysis, we would expect the following outcome. So if we conduct a PCR using a pair of primers, that is, in the 5' is part of the UBI promoter sequence, and 3' primer is part of the downstream gas coding sequence. Okay, so because it's a huge fragment, and using a normal polymerase and it could not be amplified, but the same pair of of primers in the second plasmid, it would, if there is no recombination taking place, we would have a 3.3 kb fragment. But if the krill recombinase is working, that will remove the bar gene, so we'll have a shortened fragment about 2.5 kb. If the whole thing is removed, then we will have about 561 base pair fragments. And the same thing for the first construct. If the whole thing is working, we would expect also a 476 base pair fragments. So indeed, what we observed here from the gel picture on the bottom right, we can see the PCR products. And the F1 generations, we see a shortened fragment. The parent 2, we say the expected 3.5 fragments, 3.5 kb fragment. But for all the F1 generation, we have a almost perfect 2.5 kb fragments in most of the cases, indicating the complete function of Cray recombinase, which removed the bar gene and giving rise into a shortened fragment of 2.5 kb. We also observe actually 2 expected smaller fragments, which is the product of the function of both recombination systems. But unfortunately, you can see that we still have the bigger fragment, like 2.5, that indicate that although the Cray recombinase is working perfectly, but the flip FRT is not working as what we expected. So it's probably function much less efficient. It's one of the reasons. But nonetheless, the 2 system is working, and that actually achieve what we would expected, right? So this recombined fragment has been inherited into the first, the second generation, third generation. We always say 2 expected recombinants, and actually, in some individuals, the 2 fragment has been segregated away. Okay, so from this data, it indicates that the system itself is working, but it seems that the functionality of the recombination system is really depends what system we're using. So apparently, Krelux in this system is working very well, and it seems that flip FRT system from the Saccharomyces cerevisiae from yeast did not work as what we expected. So for that, and in the final construct, we decide to switch to another system with 5C31 and ATT, which actually catalyze the one direction reaction. So we expect that this was more efficient than the flip FRT. Okay, and then considering the induction of male sterility or total sterility, we also incorporate the new technology. Besides RNA interference technology, we also trying to incorporate the genome editing approach for inducing the male sterility or total sterility, which we have we have confirmed its functionality in creeping bentgrass by transcendency in protoplast, which I'm not getting details about the procedure for this. Again, we use the guide RNA to direct to the floor leafy gene to trying to mutate the genes and achieving the modification of these particular genes. So with that, we come up with a new system, the same concept, but instead we have replaced the flip part with the 5C31 ATT system, and we also incorporate the Cas9, CRISPR-Cas9 system in addition to RNA interference technology to induce male sterility. Specifically, in the first construct, and also for the gene of interest, we use one of the genes, which is the E3 small ligase from rice, which we have tested, and that will have the capacity, when overexpressed, will enhance the plant root development, enhance plant heat tolerance, and nutrition deficiency tolerance. Okay. So with all those, we then come up with the new construct in which we have, again, very similarly, we have a ubiquitin promoter, constitutive promoter driving the downstream totos-related genes, okay, which is floor leafy. Okay. And then we have also in the staffer fragment, we have the CRE recombinase system. We have our 35S driving hygromycin resistant gene for plant transformation. We also have the Cas9 expression cassette. Okay, and we have in the end we have the RNA interference for floor leafy genes. That's the first construct. In the second construct, we have again the constitutive promoter driving the rice E3 ubiquitin ligase separated by staffer fragments. Again, the bar gene flanked by recombinant CRE target site LOXP, and then followed by 5C31, another recombinase, and the guide RNA for floor leafy. So in this case, we transform those two constructs into the creeping bentgrass. Okay, so we would expect that when crossing those two transgenic lines will produce a F1 generation in which those two constructs will be brought together, and we will have the CRE that is expressed to be functional to remove bar gene first, and then bring together the upstream UBI promoter to drive the 5C31 expression, which will then remove the whole thing there, 5C31 and guide RNA, the whole thing bring upstream ubiquitin promoter to downstream of E3 ligase gene. That's the gene of interest. And on the other side, the staffer fragment will also be removed. We have then brought together the upstream promoter to the downstream RNAi construct the floor leafy genes that will achieve male sterility. And at the same time, before this takes place, the CRISPR-Cas9 system will also supposed to work to mutate the floor leafy gene. So that has a double strategy for achieving the total sterility. So this is actually, we have the transgenic lines. We are waiting to actually fertilize those plants and for producing F1 generations. Okay, so with all that, I would like to kind of summarize what we have been trying to do in achieving the gene containment and also remove all the unnecessary DNA fragment in the final product. So first of all, the transgene containment is very important, especially when you use transgenic approach for transgenic product development. And then secondly, male sterility and total sterility are still effective for preventing transgene escape. And a dual site-specific recombination system taking advantage of the tissues, the specific action of the recombinases, and also the power of inducing total sterility or male sterility, and combine all those together can achieve both gene containment and also the so-called ideal clean final transgenic products. Right. With that, I would like to just stop here by thanking the former lab members, the postdoc and graduate students, and a lot of undergraduate students doing direct research in the lab, and also my collaborator, Also my former advisor, Tom Hodges, which is retired from Purdue University, and Dr. Albert Koch from University of Rhode Island. I think he's here in the audience. And of course, the financial support from a couple of grants from the USDA BRAC programs and student assistance by Clemson University. With that, I would stop here. Thank you for your attention. And if you have time, okay, I'll be glad to answer any questions. Yeah.
So I'm going to ask you about something that you hinted at in your slides but didn't really talk it very much, and that is removal of extraneous DNA, including vector backbone sequences. So we showed many years ago about the vector backbone sequences, other labs also. Bakir-Olker showed that chromosomal DNA from Agrobacterium could get into the plant linked to the DNA. We've also shown in unpublished data that virtually any DNA from Agrobacterium could get into a plant from other replicons. And so we're wondering, I'm wondering what your ideas are of getting these things in, because basically, I mean, it could become a regulatory nightmare, but basically anything in Agrobacterium can get into the plant.
Yeah, that's a very good question, and to be frank, I don't have an answer to that. But I think there are different strategies as far as the transformation approach is concerned, right? Agrobacterium mediated transformation probably just one of them, which is probably most popular and most effective way for plant transformation, and which brought its own problem, like what you just mentioned, not only tDNA, but also other parts of DNA and getting into the host genome. And to do that, I think it really depends on the species and it depends on the cases. Maybe additional transformation approach, bionistic approach, or any other new nanotechnology-based plant transformation technique could be used in this case to overcome this uncertainty or, regulatory nightmare, if you want. Yeah, that's, thanks. Well, I guess, yeah. Okay, so with that, I'd like to introduce our next speaker, Dr. Alison Elleman, sorry. Pronounce your name correctly, sorry, yeah. So she is a professor in the University of California, Davis, an expert in animal biotechnology. So she is going to talk about the biotechnology landscape in animal biotechnology, right, if I remember the title correctly. So with that, please welcome Dr. Alison Elleman. Yeah.
All right, good morning everyone, and we're going to switch kingdoms now and move over to animals, which last time I checked was about half of global agriculture, so thank you for including them here today. And basically give a little bit of background to the global regulatory landscape for animal biotechnology. A lot of this is work of my postdoc in terms of reviewing what's going on around the world, and that is, I'm just giving her credit because sometimes they do all the work and then we get up here and give the talk. So I always like to start with this slide because obviously we can make tremendous genetic changes to our domestic livestock and pet populations just based on naturally occurring mutations that exist in the genome. And the reason that these animals no longer look like a wolf is because certain breeders thought that a poodle was a beautiful animal and they went for that, and someone else thought a bulldog was great. You may not think this is an improvement over the wolf, but these breeders did, and some people select them to live with their children, and probably if they still had the phenotype of a wolf, we wouldn't be so comfortable leaving our kids around them because they would be eaten. And so I think these types of breeding is what most of our genetic improvement programs is based upon, but as we know, around about 40 years ago, we had the quote-unquote modern biotechnologies that came into existence, and this is kind of an abbreviated timeline, if you will, of the first genetically engineered animals that were produced, or livestock specifically, back in 40 years ago in 1985. And it's a little bit sad for me. I wish someone had shown me this when I was an undergrad, because I probably wouldn't have pursued the career I did, how little progress has been made since then. So you can actually name these animals, or at least I can. Looking at them, I see the Aqua Advantage salmon there, the fast-growing salmon produced. I can see Herman, the first animal to produce a pharmaceutical product in his in the mammary glands of his offspring. And then, of course, the infamous Dolly, the somatic cell nuclear transfer cloned sheep that came around back in the, well, just before 2000. And then we have the blue line there that's indicative of when genome editing kind of came into play about a decade ago. And I think, sadly, although we've had previous talks talking about the millions of farmers globally growing genetically engineered crops, be they insect protected or herbicide tolerant, we've only ever had 2 food use approvals for genetically engineered animals in 40 years. And that's pretty pathetic. One of them was the Aqua Advantage salmon that was actually in the published literature in 2001 and got its regulatory approval basically a couple of years ago. So what does that tell developers? What does that tell industry? Don't bother. And that's what industry did. They didn't bother. They pulled back their money. There was no funding. USDA ceased funding genetically engineered animal research because they said there's no point funding research that's never going to go to market. And basically the field just withered and died. And then along came genome editing and opened up new opportunities, because the thinking was that, well, this is going to be regulated very differently to transgenic organisms, because there is often no foreign DNA inserted, and so there is no transgenic construct to trigger GMO regulation. So let's talk about genome editing. I am not, for this audience, going to discuss what it is. I only put this up because I want to distinguish between the non-homology end-joining pathway, the SDN1 pathway, which I'm going to call the left-hand side of the repair pathway here, versus the knock-in or the homology-directed repair pathway, where a donor template is provided. And the reason I make that distinction is, from a regulatory perspective, that's really important. And so from a biological perspective, it's who cares? But from a regulatory perspective, they're in fundamentally different camps. And I think it's frustrating that as biologists, we're trying to work around the regulatory system to achieve our aims, even if it's not often the most direct approach to achieve our aims. And so we're dancing around this not fit for purpose regulatory system to try to improve the genetics of plants and animals, and going to great lengths to remove, foreign DNA or worry about off-target mutations, when I would argue, well, what is the hazard we're trying to avoid here? Must be very, very dangerous to spend so much time doing it. I have a really hard time coming up with one, to be frank. So there's that. Okay, so genome editing is different. How we do it in animals is different to plants. So no cell wall in animals, just to remind you, basic biology. So we are somewhat fortunate in that we can introduce our editing reagents in an RNP, or ribonucleic protein complex. We don't often, most of the time, actually integrate transgenic plasmids the same way that plants have to, and so this removing of the cassette and that type of discussion is moot to some extent. So the way that we have to do this, obviously, as an animal breeder, I don't really care about the somatic cells of my animal. I only want to target that germline. So they have to pass this on from generation to generation. And there's 2 basic approaches the way that this is done. One of them is introducing the editing reagents into a 1-cell zygote, shown there, just after the sperm has fertilized the egg, before the first cell division. And ideally, you want to introduce those editing reagents and produce a homozygous, non-mosaic animal. That is an animal where both alleles have been edited for whatever edit you're after, and it's done before that first cell division, so that that animal has the same genotype in all of its cells, including, obviously, the entire germline that's derived from a 1-cell zygote. The other way that this is done, and I don't show here, is you would do your editing in a cell line. You would get your homozygous edit, confirm everything is correct, and then you take that nucleus and do somatic cell nuclear transfer cloning a la Dolly to produce a homozygous non-mosaic animal. So those are kind of the two approaches. That latter approach only really works well or it's open to mammals. It's not really open to chickens, for example, because of the way that those are edited. So that's the kind of the differences between our transformation pipeline and plant transformation pipeline. So we did this review of 195 peer-reviewed publications that were working on genome editing for agricultural applications. So divorced from the biomedical applications and transplanting hearts from pigs to humans, all of that out, just food production. That's my interest. I'm an agricultural scientist. And basically we looked at what people are doing in terms of editing, and as you can see, good old CRISPR-Cas9 is king at the moment. In 10 years' time, it'll be some other one, no doubt, but at the moment, that's what's being used. A little bit of the TALENs and zinc fingers, but they tend to be older papers, just because that's a more complicated technology to use, and then a couple of others. And DNA-based editing is really at its very, very infancy in terms of animals. We just haven't got high efficiencies to where it warrants using that. And if we look at how these animals were being made, about 30% of the applications were edited in cell lines followed by cloning. And that's often done when there's a requirement for HDR repair. So HDR repair requires basically a replicating cell. That's very quiescent in a zygote. That is a one-cell embryo, and so knockouts are relatively easy in zygotes, but almost impossible to get a knock-in, and so that you need a dividing cell line, and so most of the knock-ins have been done that way, followed by somatic cell nuclear transfer cloning. That's mammals. About 60% have been knockouts in developing embryos. As I said, that pathway, NHEJ, is quite easy, relatively speaking, in embryos, and so that's been that approach. And then the other 9% are poultry, and it's a long story, but chickens, when the egg is laid, is many thousands of cells. There's no way to get to a one-cell zygote in a chicken, and so they have to actually edit primordial germ cells and then put that into the developing chicken, and so that's a little bit different the way they do it. But interestingly, the majority of the applications to date, 75%, have been SDN1 knockouts, NHEJ. And there's two reasons for that. One, it's relatively easy to do, and two, it removes you often from regulatory GMO regulation. And again, so we're going after this in part because the regulatory pathway is too complicated once you get into the GMO regulations. And that is a little bit insane, because there are actually very valid and legitimate reasons why you would want to use a template to introduce a useful allele, and to not be able to do that because of the potential for triggering GMO regulations isn't a very scientifically rational approach to making that decision. So why? Why are we editing? Well, we haven't changed the objectives of our breeding programs. We're still interested in the same traits that are in our breeding objectives to start with. So if we look at the breeding objectives of animal agriculture, it's going to be not only the product they're producing, but we want healthy, fertile, robust, long-lived, all of the things that you want in plant breeding. And so this breaks it down by different animal classes. And so ruminants are obviously cows, sheep, goats, four-stomached creatures. Then the monogastric mammals are pigs and rabbits, and then the aquatic animals are obviously fish. Avian is self-explanatory, and then insects. And what you see at the bottom, not surprisingly, yield is a big trait. Well, that doesn't surprise me. That is an important component for breeding objectives. There is this one gene called myostatin, where you can knock it out and get this very, very dramatically increased muscle yield trait, and we see that in the Belgian blue cattle. And that one is like a favorite for every lab to do, because you get these really muscular looking animals, whether it's a sheep or a cow or a fish. And so a lot of that is really developmental research or basic research that's not actually going to market. Then you see reproduction is number 2. There we're targeting traits like single sex offspring, so layers, we don't need males because they don't lay eggs. Dairy, we don't need males because they don't produce milk, and so there are reasons why you want certain certain sexes, or you want infertile animals for transgene containment or genetically edited containment. If you have an edited fish that's infertile, then you don't have any issues with potential interbreeding with wild populations. Biotic stress is disease resistance, very big target, and then allergenicity, removing allergens. Colour is important for particularly fish, so koi carp and that type of thing, the colour is important. Other is things like welfare traits, like removing the horns off cattle, and then at the top there is abiotic stress, which would be heat tolerance and coping with a changing environment. So let's have a look at some of the SDN1 applications, and so these would be knockouts, so knockouts of products that are allergenic in milk, for example. Thermotolerance is a particular mutation in a prolactin receptor that gives animals a slick phenotype. That's a coat variant that allows their body temperature to remain 1 degree Celsius cooler than their non-slick counterparts. It's a naturally occurring allele that occurs in many tropical breeds that you could introduce, for example, into the more temperate breeds to cope with an increasingly warm environment. Sex ratio skew, in the case of pigs, you don't really want the males because they have to be, once they reach sexual maturity, they give a nasty flavour to the meat. It's called boar taint, and so they have to be castrated to remove that, and now that's been banned in Europe. You're not allowed to castrate pigs in Europe, and so they're killing the pigs before they reach puberty, but ideally you would like them to actually be harvested at a later date, but not have this nasty sex ratio skew, so in that case you'd like all females. Unwanted development, you could knock out the boar taint pathway. Increased yield, the myostatin knockout I mentioned earlier, and disease susceptibility, and there's one example that has been approved that I'll get to later, which is porcine reproductive and respiratory syndrome virus, which is a devastating disease of pigs that's endemic here in the US and in Europe and through most of the world, and basically there's a knockout of a single exon of a gene that renders those pigs no longer susceptible to that particular virus. I'm a cattle geneticist, and so a lot of my examples and pictures of cows, sorry, just have to put them up there. So here's a cow that's got the beta-lactoglobulin knockout. This has been done in New Zealand. The flag is indicative of where this work is going on. Here's an example of the SLIC mutation I mentioned a minute ago. So the cow on the left has the SLIC phenotype, so you can see its coat is kind of shinier and not as hairy as the non-slick animal, and basically, as I said, keeps them cooler. What might we introduce? So now we're moving into the homology-directed repair pathway. So often what we're doing here is actually introducing an allele from a different breed into the elite germplasm of the breed we're interested in. So we're not necessarily bringing in transgenic DNA from a different species, but rather moving alleles between breeds without having to crossbreed. So basically you can bring in the targeted trait without the linkage drag associated with basically mating a animal of that breed with your animal and getting the trait you're after. So again, our breeding objectives haven't changed. We'd like animals that don't get sick. We'd like animals that don't grow horns, because you normally have to cut them off, and that's quite an animal welfare issue, and farmers don't particularly enjoy it, neither do the animals. Thermotolerance, maybe changing the coat colour to be not so absorbing of heat, altering the food quality, and potentially sex ratio skew, and I mentioned that earlier about the layer chickens. We really don't want to hatch boy chickens into the layer industry. So here's some examples. This is work out of China to try to produce tuberculosis-resistant cattle. This is a really interesting example out of the Meat Animal Research Center in the United States, where the researchers had a hypothesis that if they altered 18 base pairs in this one particular gene, CD46, and they substituted 18 base pairs with a different 18 base pairs that encodes for 6 different amino acids than what the wild-type protein does, that would render an animal that's no longer susceptible to bovine viral diarrhea virus, which is BVD, which is a major viral pathogen resulting in respiratory disease. And so basically what they did is they targeted the cell line from a gear. That's the animal shown on the bottom left. That's what they normally look like. That's not an edited result, that's just what they look like. And basically they're an animal that's tropically adapted and is used widely in tropically adapted countries. And they took the cell line, edited that cell line to introduce that guide RNA. So this would have been a template guided repair, and substituted it so that the protein no longer was the wild type, but it had instead this basically ALP-TFS, which is the amino acid encoding by that 18 base pair change. And they got a calf shown there after cloning it, and when they challenged that calf to this disease, it was less susceptible than a wild type animal. That's the wording they used in their paper, so they didn't say resistant, but less susceptible. So that's a really nice example of really directed evolution, I guess, or for want of a better, but basically having a hypothesis, being able to achieve it, and then seeing if that does the phenotype that you want it to. This is an example from New Zealand where they basically produce the calf on the left that is got this lighter colour coat that wouldn't absorb heat so well as black and white Holsteins normally would, and so again for coping with a warming environment. This is work that my lab was associated with, and it's gene-edited polled calves, so basically bringing in allele from the Celtic breeds of beef cattle like Angus that don't grow horns, so there's a naturally occurring mutation. It's a 202 base pair insertion upstream of the promoter. We have no idea how it works, but it stops the horns from growing. And if you introduce that 202 base pair allele into Holsteins, which normally grow horns, you will have a polled animal, an animal that doesn't grow horns. So you don't need to burn it off anymore, which is what is normally done to protect the animals and to protect their human handlers. This is getting old now. It's almost a decade ago that this was first done. And then this is an example, again, from Excelligen, which is a company that's working in this space. And what they're doing here is starting to stack traits, and so they're interested in taking a Holstein, it's not really a Holstein, it's an adapted animal for the tropics, and they've introduced the slick allele into it, so the prolactin receptor, and then in cell lines using what's called bovine embryonic stem cells, they've introduced two edits in two genes, FDX2 and DHRS4, which they hypothesize will make those animals resistant to sleeping sickness or trypanosomiasis, which really limits where cattle can be used in Africa, and it's a zoonotic disease. It kills not only cows but also humans, and they've done all of that in a cell line and then cloned that to produce this line. And this is actually work that's funded by Bill and Melinda Gates, and they've got males and females of these animals, with the intended market being in the tropics and subtropical zones to have dairy cattle that are better able to produce milk but also cope with the environment there. So where is this work being done? And this is a really interesting slide. So this is basically breaking down the authorship of the 195 papers that we identified in the literature. And if you're interested, we've put all of this data into this database that's shown here at the top. And it has all of the papers that we reviewed, and you can kind of dig down into it. But what really stood out is that over half of the papers that are in the peer-reviewed literatures, this is not in development but just basic research, are out of China. And you can see that they're working in all of the different categories of animals there, followed by the United States there, and then Japan, South Korea, Norway, that big seven, that's salmon basically, trying to make infertile salmon to basically contain genetically improved salmon lines to prevent interbreeding with Atlantic salmon. And then there's a little smattering, I guess, for want of a better expression, in all the other countries. So obviously a lot of work getting done in China on this, and a lot of investment by their government, which tells me that they're interested in using this eventually in their food supply, because why else would you invest in this? And so I think that's an interesting one. So if you want to go to that gene-edited animal database, so this is, for an example, if you go there, you can have a look at what different species are being edited. And I'm Australian, so I'm going to do the example of sheep, because they're a lot of sheep in Australia. And so if I click on sheep, then I'll go into this table here, which is all of the edited papers that are on sheep. And if I want to, you can click on any of these, view details, and it'll drill down into what what's been edited, what for, how did they do it, did they use cloning, did they use editing. And the idea behind this whole database is to be transparent about what's going on globally. There was a lot of pushback against secretive GMO, Monsanto evil types. So this is just, there it is, this is what's happening around the world, so you can go check it out. So what's important here is these are ones that have not undergone a regulatory process, and I'm going to go into regulations now, and I'm a molecular geneticist. I really don't give a rip about regulations, but my entire life has been consumed by regulations. And so, unfortunately, I've become an expert in regulations as it relates to genome editing, not because I want to, but because it really has held back the field of genetic engineering my entire career, and I worry it'll do the same for genome editing. So let's have a look at regulations. So this really breaks down the nut of the problem. So as has been alluded to yesterday, natural mutations and mutagenesis using radiation, up the top there in red, are not treated differently to conventional breeding. They are actually the basis of conventional breeding. Then down the bottom there, if you have a transgenic recombinant DNA insertion, that has historically been the trigger for regulating. Well, with editing, there's kind of all this in-between stuff, right? So there's single nucleotide changes, there's little SNPs and indels. Then there's potentially using a template that's a short template, that's an allele that already exists in your species. Or you could use a template that contains transgenic DNA, and you could produce a quote-unquote GMO if you use a foreign DNA as your definition. So how are we going to regulate this new technology? And so what I think is fair to say is that the bar has kind of been made there by most countries that have looked at it. So if you have foreign DNA under the Cartagena Protocol of definition of recombinant, then you'll be treated as a GMO. So if you've got a transgenic repair template that's got foreign DNA, then you'll be regulated as a GMO. If you could have created it using conventional breeding and there's no foreign DNA, then you are not going to be treated any differently. And it's really this kind of in between that long repair transgenic, cisgenic template and the transgenic repair template is where it gets a bit dodgy. So that resistant animal I showed you where they introduced 18 base pairs to do the 6 amino acids, that's novel DNA. Is that foreign? Well, it's not foreign. It was created in some researcher's head, but it hasn't existed in that germplasm before. So does that make it transgenic? Or could that have occurred conventional? Well, could have eventually, like all mutations can happen. So it becomes a little bit dodgy there. And then some countries, and I'll point out good old Australia here, just the way their regulations are written means that if you use a template, doesn't matter if it's cisgenic or it just introduces a 1 base pair change, the fact you've used a template is what triggers GMO regulation in that country. And so we have the global regulatory landscape for products of genome editing shown here, and I get very frustrated when it gets put up, this is the landscape for biotechnology, without saying either plant biotechnology, if that's what's being talked about, because actually the regulations are very different in some countries between plants and animals for no rational reason, but it doesn't, when we say, oh, it's easy to get through, GM or getting edited plants into the market in America, that's absolutely not true for animals, and they are half of agriculture. So if we're going to talk about it, let's talk about the products of genome editing globally, or narrow it down to plant biotechnology or animal biotechnology. So what's shown here in the green is countries that have exclusions, basically for small edits that don't contain transgenic DNA. And this was really led by Brazil, and I give them a lot of credit for basically taking the bull by the horns and coming out of the gate early with, if there's no transgenic DNA, it's not covered by the Cartagena Protocol, we are not going to treat that differently to conventional breeding. And many countries have followed suit in that regard, and this has to be updated because Africa is very much going after that Brazilian model. But then you'll see some countries that have the weird stripy stuff, including America, and I'll get to that in a minute, because yes, they have the approach for plants, but they have a very different approach for animals, and that is regulating all edits, irrespective of the nature, as new animal drugs. Then you have some countries like South Africa there that's just like, I don't care what you did, if you use genome editing, you are a GMO and you're going to be regulated like a GMO. Then you've got Europe that's making up their mind, still kind of in the yellow bit. And then you've got like Australia with the stripes there, that the reason they are in stripes is because of that template is what triggers GMO regulation. And so it varies around the world, but the problem is, because we trade animal products globally, if there's one country that's like, no, this is illegal, then either we stop doing trade with them, which is not easy, or we say, okay, we're going to let the most precautionary, prohibitive approach dictate global use of innovation. And that really concerns me, because they tend to be rich, wealthy countries that have the most precautionary approach, and that affects the ability of developing nations to use the best innovation in agriculture. And that is just, I don't know, regulatory colonialism, I guess, for want of a better expression. It's just wrong. That, unfortunately, is the outcome, though, because we do need to trade animal products. And if we can't trade because we've got a knockout in Brazil with Europe, then basically Brazil can't use that knockout technology. Or they can choose to just isolate those countries and not trade with them. Maybe that wouldn't be a bad idea, but I'm not one to make that judgment. So if we go and have a look at the ones that have undergone a regulatory process, this is the list. And as I mentioned, there's only been 2 approved genetically engineered products in animal agriculture in 40 years. This is already the list of gene-edited products that have been given a regulatory determination. Notice I didn't say approval, a regulatory determination where they've been judged to be non-GMO. It's kind of analogous to the am I regulated discussion we had yesterday. So if you're not got transgenic DNA or foreign DNA, in many jurisdictions you're not going to be treated differently to conventional breeding. And so you don't have to get an approval because you're traditional. There's nothing there to regulate. And so this is what it looks like, and you'll notice there's a lot of South American presence here, and because their regulatory system is more fit for purpose, what they're seeing is they're getting products through. And that's good. They're letting products through from small developers and often local developers. And so this is an example of Argentina, where even 5 years ago already, they were making non-GMO determinations for slick cattle. So that's the prolactin receptor that gives it that slick coat. And also for a animal that would be slick and polled, so no horns, heat-tolerant Holstein for their dairy operations in Argentina, and they basically, their determination was there's no foreign DNA, so there's nothing to regulate, so you can go to market if you want to. Doesn't necessarily mean those companies have gone to market, but they could. There's nothing stopping them. This is Brazil, and so basically they have said similar determinations for a myostatin knockout, so that Nelore animal on the right there with the big muscles, he has got it's a partial knockout of myostatin, so increases muscle yield, which is an important trait. No foreign DNA, just knocked out myostatin gene, not treated differently to conventional breeding. And then those animals at the bottom there are Angus that have the slick allele introduced, and they have been given non-GMO determination in 2021 by Brazil. Now we get to the US, and the FDA guidance on this is basically says, if you're editing mice, rats, rodents, we don't expect you to come and approach the FDA. So that way they get all the medical community off their back, because they can keep using and doing their research and no one's going to impact them. If, however, you're doing food animals, then there's category 2, and that is what I mentioned yesterday, products that we don't expect developers to submit an application of approval if it's a low-risk product. So if you're introducing an allele that already exists in the food, there's no novel DNA or protein that could be, allergenic or toxic, then we could give you a low-risk determination, which is what's called enforcement discretion, that allows those products to come to market. And that's what happened back in 2022 with those 2 slick animals. That's unique for those 2 animals, that determination. If I make a slick animal, I'd have to go through a separate regulatory decision on that. And these animals, they're milk, meat, and gametes could all enter the food supply, or, well, the gametes wouldn't, but they could all enter commerce because they've been given a regulatory enforcement discretion decision. And then finally is category 3, products for which a full new animal drug approval is going to be required. And that's basically what happened with this. You would have maybe heard about this. The porcine reproductive respiratory syndrome pigs were given a full approval in April of 2025 in the United States. Basically, pigs are a 4-way cross, so they had to edit in the homozygous condition the gene that affects this disease resistance to PRRS in all 4 of those lines, both males and females, so multiple founders in multiple lines, all had to go through the regulatory review process, multi-million-dollar, multi-year process, but this is a large multinational corporation, so they could afford this. Are they the only ones we're going to allow to bring products to market, these large companies, or are we going to have an approach that allows smaller companies to play? So this is where we are in terms of products that have undergone a regulatory review, low-risk approval or approval determination, and you can see, again, that South America is leading the charge there, and you can see US at the bottom with the SLIC and the PRRS resistance approval. I'm going to just focus for a minute on Japan. So they have 3 fish approved there, so the tiger bream, the puffer fish, and the flounder. All of them are fast-growing, getting to market weight in half the time of conventional fish. And actually, one of them is a myostatin knockout, ironically enough, myostatin bream. And basically, what happened was that a researcher developed them, and then he brought them to market himself, formed a little company. And it was anticipated that there would be a lot of pushback against this because previous GMO controversies in Japan were driven by major news companies and consumer groups saying this is really dangerous and you need to be fearful of this technology. And then they did a poll of the public and said, well, what would it take for you to accept genome-edited animals? And they're like, well, we want really, really strict, tight regulations for genome-edited animals to reduce the risk as close to 0 as possible, rather than scientifically proven regulations and technically reasonable. But that's not what Japan did. They said, you know what, if there's no novel DNA, we're not going to treat it differently to conventional breeding, and that allowed these products to come to market. And I think what's interesting is, even though there were some social actions and groups were trying to get people, up in arms around this, it just didn't catch, didn't take off. And basically those products, and the media coverage was mostly positive. Partly, I think, because it was a local university professor developing fish. It wasn't, Monsanto, a large multinational company. It was, Dr. Jones. And so I see editing as a cherry on top of the breeding sundae. So we're still going to have to do all of the things that traditional breeding programs do. And in the case of animals, that includes all of the technologies under genome editing. But what it enables us to do is introduce useful alleles without linkage drag. And that's its main superpower, in my opinion. But, as I have been waxing on about for years now, if we do not have product-based trigger for the regulations and sensible, risk-proportionate regulations, we're going to have problems globally commercializing it. So genome editing offers an approach to introduce useful variation. Scaling edits to commercial livestock populations is not for the weak of heart. It's going to be expensive because of our breeding design of our elite nucleus populations. Regulators in many countries are not treating edits that could have been achieved using conventional breeding differently to conventional breeding. That's not true in America and Australia. If you have a template, you're going to be regulated as a GMO. And in my opinion, the fate of this technology is going to be very much dependent on getting some type of harmonization between the different countries in their approach to regulating this. And if there are blocks like the European Union that are going to treat this as a GMO, that's going to be kind of a de facto prohibition on using this technology for their trading partners. And that's what happened with genetic engineering. Africa was nervous to use it because of the potentially jeopardizing the European markets, but it also has this perverse effect of preventing useful technologies being used in developing countries because they're worried about their markets in the first world. And so thank you for your invitation here. I like this slide because to me, editing, biotech over on the right there, complements our advanced reproductive technologies like cloning and embryo transfer and genomic selection to come together to synergistically accelerate the rate of genetic gain in our domestic animal breeding programs. So with that, thank you very much.
Thank you. Just curious, why some example of editing in animals is considered as a drug by FDA here in the US?
Beats me. No, so technically the legal definition of a drug under the Food, Drug and Cosmetic Act is an article that alters the form or function of an animal. So they've interpreted that to mean that genetic variation alters the form or function of an animal. And it undoubtedly does. Like, the reason that you and I don't look the same is we have genetic variations between us, right? So it alters our form or function. But that's true of all DNA. And so if that's your definition of a drug, then we're all drugs. And every animal we ever produce is a drug. So to me, it's a not fit for purpose definition. That definition was introduced, actually, ironically enough, when there was a lot of charlatans selling garbage back in the 1930s. That's actually what introduced the Food, Drug and Cosmetic Act, was you have to show a drug is safe and effective. That was the genesis of that act. And it's just ironic to me at this juncture in history with what's going on with the natural foods industry that, like, that's all getting questioned now. But there was no definition back then of what DNA was, because DNA hadn't actually even been discovered in the 30s. And so there's no way that the founders or the people that wrote that legislation thought, oh, this should cover genetic variations. And it's insane to me.
Thank you.
Wonderful talk, Allison. I know way more about plant biotechnology than animal. And in the plant biotech industry, there are very strong considerations that have implications on regulatory policies, and these are associated with the grain movement across the globe and export markets and things like that. And for example, if an article, a biotech product in major crop is approved in the US, for example, but not approved in other countries, that creates very, very significant challenges introducing it, right? Because it can be found in unapproved market, and then with all the implications. Any considerations in the animal field like that? Is animal trade globally going on to the extent that it also imposes restrictions?
100%. So, I mean, it depends a little bit on the application. So those fish in Japan, they've only been approved in Japan, and they're servicing a local market. So they're not entering trade in the same way that, for example, I don't know, soy is, for example. With regards to large multinational corporations playing in this space, and I'm talking specifically here Genus with that PRRS-resistant pig, they are very cognizant of the fact that they need approvals in all of their major export markets, and so they are diligently working to get, you saw they've already got it in Brazil, Colombia, Dominican Republic, America. They're trying to get China, they're trying to get the major partners. Interestingly, they're not bothering with Europe. So they're a UK-based company, kind of Europe adjacent, and they just realised it's not even worth their time. They'll just keep getting the sick pigs. And I find that ironic, because they have this green policy in the EU where they want to try to reduce the environmental footprint of agriculture, but they're actively prohibiting the use of disease-resistant animals, which, there's nothing less sustainable than a dead animal. And so we have this perverse situation where it's just like, okay, we accept that Europe's just going to be impossible, we'll just keep giving them the sick pigs. Or not sick, but susceptible. And they will keep getting sick and dying. And the rest of the world will have access to these pigs that don't get sick, or at least the rest of the world that has regulatory approvals. So absolutely, the large multinationals are aware of that. Now, when it gets to the smaller companies that are scrapping along, trying to make a living, and I mentioned one where there's 2 cows that have an approval with Exalogen, and there are animals on properties that I'm aware of, as in ranchers' properties where they've got a particular edit that's been done. What's going to be the constraint on those guys? Who knows? Especially for things that are being regulatory enforcement discretion, because there's nothing stopping that meat, milk, and gametes from going into commercial production. And what is going to be the hold over that? And it's like there isn't something analogous to what there is in plants, because the industry structure of especially beef cattle is so dispersed. There's no large Monsanto-ish kind of an entity. There's some AI, as in artificial insemination companies that are large, but in beef cattle, it's mostly ranchers that are breeding their own animals or buying it from their mates and stuff. And it's going to be a lot harder to get your arms around that. And you could argue, well, in the case of slick allele, that's existing in conventional breeding. Even if you have a DNA test, you're not going to be able to differentiate whether that was naturally bred in or it came from. So if you can't detect it, then it's a silly regulation and people will ignore it.
Thank you.
If not, let's thank all the speakers, including me.